So please.
Okay, Darrell Lund. This is a question for Barbara. Marijuana. When we see the national news media go out to Colorado and talk to distributors of marijuana, they make all kinds of claims. Is this an issue that is not going to come before FDA simply because it's not legal? Or can Coloradoans make up whatever they want to say about it?
I'm not sure I'm the right person to answer that question. If we just call it substance acts, let's not, so it is legal in one state. The claims, I mean, if you were simply looking at it from a simple food perspective, number one, safety. You've got to deal with safety before you deal with anything else. And number two, if they're unapproved health claims, then that means they are unapproved drug claims. They are unapproved new drug claims. That's the authority FDA uses when it goes after inappropriate claims on products. But because of the nature of the substance, my guess is you're on the drug side of the agency or you're with drug enforcement agency. I don't know that it necessarily falls under FDA's purview or even CFSAN's purview. I don't know, has NIH had any experience with it?
Rodney, you want to hit on that question? I didn't see that one.
I don't think I have much to add.
Okay. Thank you, Tim.
So, Tim Work from Nestle. Cindy, you started out with a really nice overview, and you showed that some of the changes of different patterns of microbiota and sequencing changes metabolites. You were talking about polyphenol metabolites and butyrate production and all these other things. So here's a bit of a provocative question. How do we talk about normalizing a microbiota? And Dr. Dieter suggested about completing and having a completed microbiota. So if we're really talking about affecting change from a dysbiotic state to a normal biotic state, whatever that might be, that's not a health claim. That's not a disease claim. That's a metabolic normalization claim. How do we deal with that?
He's asking you the question.
She introduced it. You got the regulatory side.
I think you're going to have to answer.
Because I think this is really what we are as nutritionists. We talk about metabolic states so much of the time, and it includes the whole gamut of nutrition, but it ultimately results in your metabolic condition.
Which is, again, why I put that slide in at the end. I figured it might provoke some discussion. That it really starts to beg the question, and I think, Tim, this is a point you've made in the past before. When you have people in a disease state, they may have different nutrient needs. And I think a fair question in looking at that is, we know that there's a variation in nutrient requirements. The RDAs have reflected that since they began. So what is a within normal healthy population variation in nutrient requirement? And when does it step outside of that normal range, but it's still a nutrient requirement? It's not a drug. It just means because of your metabolic state, your nutrient needs are somewhat different. We certainly recognize that in certain cases, like a burn patient, a surgical patient, we might see that. So to me, when I was at the agency, I'm not sure I would have asked this question, but I will ask the question now that how do we deal with that state? And what role should food and nutrition have in managing disease states? Is that something we simply leave to the drug side, or is there a role that food and food products should be playing in that management? And to me, it's especially relevant when we start looking at some of the lifelong diseases that people may have.
This isn't quite an answer to your question, but it also comes back to Connie's question earlier. And I think it really comes down to is what is the metabolic capacity, like you were saying, and when there's dysbiosis, what metabolic functions, microbial metabolic functions are missing, and then what do we need to normalize it? And part of the problem is we really don't know that yet. So much of the research has really been on is who's normally there and who's not there. And they realize that's not the important question. So I think that we need to kind of step back, let the science catch up, and then be able to go from there. I mean, people talk about some of the studies with probiotics, but you really look at the probiotic literature, and so many of the studies are using different doses of probiotics, different species, different dosing regimens, different vehicles, that it's very difficult to compare the studies and be able to look at a totality of evidence to really say that it's having biological effects. There is definitely evidence that probiotics are beneficial in certain situations, but nobody's really looked at what's the baseline microbiota to begin with. What's their baseline metabolic capacity? Is there something missing that the probiotics are then giving them that then shows the beneficial effects? And I think we need to relook at how we're doing the science.
But I wouldn't even limit it to the probiotics, but because when you showed your Atkins-type diet, and you said those people might be butyrate deficient, for lack of a better word, how do you know then what the normal amount of gut-generated butyrate should be? Because then it's not a nutrient, and then it's a prebiotic to generate a metabolite in the intestinal tract, which may be beneficial for preventing colon cancer, who knows? How about Dr. Dieter, do you have a thought on that?
Just that I think it's also important to consider the life stage, because the microbiota are going to change. A healthy microbiota in a 3-day-old newborn is going to be different than that for a 6-year-old child that's healthy, and then middle-aged adult and elderly. So considering life stage, what you'd like to see in terms of diversity mix and metabolite production is really important. But I think those metabolites are a very important factor, and balancing those and understanding how to have the bacteria in place and the nutrients that are going to enable desirable metabolite production as well is really important. Derek McFabe up in Canada has shown that he can either put in the microbes or put in the short-chain fatty acid metabolites, and he can make mice and rats OCD just with the metabolites. So control over those and balance over those is really important, and I think it's diet plus microbial seeding nurturing that need to go together in the end.
Great, thank you.
Thanks, Tim. Sticking with the probiotics for a moment, what do we know about them sticking around? Someone, let's give an example of infants, children, middle-aged adults taking probiotics and then discontinuing. Do they persist? I'll start with Cindy. You want to hit on that first?
Well, the general belief is that they don't persist, that they're only there for as long as you give them. But I think that's somewhat of a naive view. If you go to the area of fecal microbial transplants for C. diff infection, they do stay around. So when is it a probiotic and when is it a MALU? I mean, part of the reason why there's been so much benefit of fecal microbial transplants in C. diff is because it's a condition where it's so dysbiotic and so lack of diversity, so few microorganisms there, that it's a different environment. So I think that the general consensus that they don't stick around is probably naive. And probably for most individuals that are healthy, they don't stick around. But in certain disease conditions, and depending on the rest of your microbiota, it probably varies. And if you have an environment that is receptive for them in certain individuals, they probably do. But we really don't know where in the spectrum everyone fits.
Thank you. Phil, please.
To follow up on that, I think most people, and if you're healthy and you're above a certain age, the probiotics only stay around for a period of time, and if you stop them, you can't detect them, even with molecular molecular techniques, so they don't colonize well. But in the younger the human and the animal, the more there is the chance they will persist. And it does relate to diversity. So there's one study with a commercial probiotic product given to a grandchild of an investigator, where the organism persisted for months to years after. And that might be a good thing, but the worry is maybe that's not a good thing. And the impact it has on the host for the long term, in particular the immune system, that's what needs to be studied. So I think there's some anxiety about giving probiotics when the niche is relatively empty, like in the newborn period, or perhaps even for recurrent C. diff, especially in a young person. And, most of the time these fecal transplants are in the very elderly, where the odds are you're not going to live another 40 or 50 years. But in a young person with recurrent C. diff, whole different kettle of fish.
I could jump in on that.
Please, Rodney.
Thanks. For the immune system, there is some evidence, particularly in rodent models, that there are narrow windows in early development where your microbial interactions are most likely to be effective. Miss the window, you may actually not have the desired immune effect that you think you're getting. So that's, again, a counter argument to going into exiled dysbiosis and acting sooner rather than later. And the reason is, again, you can still benefit from probiotics later. You may miss the chance to actually co-mature with the immune system and avoid clones that are going to give rise to autoimmunity and inflammatory disease.
Thank you.
Well, let me first of all thank all of our speakers for wonderful talks. Are we any closer to being predictive about when we monitor the microbiome for disease states? Similar to Cliff the dog, who can determine C. difficile before it actually happens? And I guess this is to Cindy, Phil, Rodney.
Well, I mentioned this study that's going on in Canada and the United States called the GEM study, which is looking at the siblings of probans who have IBD. And so the researchers are looking at the composition of the microbiome and the genome of the host to see which are they both predictors? Is one better than the other? Are the two better than either one alone? And what's clear is that the human genome alone isn't good enough, because there are polymorphisms, sure, there are 160 genes associated with IBD, but it still only accounts for 20% of the risk of developing IBD. So that's not a biomarker. That's not a biomarker, even though you can sequence the whole genome or the whole exome. So what you need is something on top of that. The evidence so far isn't compelling that the microbiome by itself is going to be enough. So the final word isn't in. The study's still being undertaken. The numbers aren't big enough. So high quality study is really required. And the interim analysis is the microbiome by itself is not a predictor. So it's going to be a more complex analysis. That's for the one example of IBD that I know about that's been undertaken.
And I'd say that the 3 integrative human microbiome projects are really similar, that they'll get an idea of what's most important, whether it be the environment, including diet, whether it's genetics, whether it's the microbiome, and I think it's also going to show that it really is the interaction of all 3.
Is there some scientific expertise that is lacking in this whole mix? And that relates to how do we analyze all this data that we're generating? And I think at the program committee we talked last year anyways about big data sets and how we analyze those big data sets. And then I guess that translates into the return on the investment, because I'm hoping that this has more promise than the Human Genome Project, where, when that first was, I guess, the results were coming out, there were all these promises.
So the question about, it's been raised in the context of missing hereditability, and is it the microbiome, or I think there is one other part that needs to be looked at, and that's the exposome. So you are exposed to a whole bunch of other things that impact on the composition of the microbiome and how your genes respond. And individual asked me during the break about environmental exposures and why aren't we looking at their impact on metabolomics. So It's true that we've looked at microbial composition, but we haven't looked at environmental exposures at varying ages, which I think is also really critical, and how that impacts on the composition of the microbiome. So maybe some of the NIH-funded studies will help address that. We're certainly trying to support environmental exposures and their impact on health and disease in Canadian research.
So in response to what you just said, the three IHMP are looking at environmental exposures too and bringing that into it. But getting to the question of big data, one of the big things that NIH investigators are looking at is how do you integrate all the different types of omics and how do you use that? How do you pull together the metagenomics with the metatranscriptomics, the metaproteomics, the metametabolomics, and how do you put it all together and how do you integrate it? And it's one of the questions that some investigators are starting to address, and particularly the Microbiome Quality Working Group is trying to look at how do you put all of these together and handle it bioinformatically. So people are working on it to come.
Great. Rodney, did you have any comment on the skill sets needed?
No, I think the presenters have covered the same kind of terrain that I would advocate.
Okay.
It's just to pay it. The only thing I guess I'd add is pay attention to the age, the physiological state, and the signals that are required. There's even some evidence in rodents, again, that there can be species specificity for microbes delivering a signal in the right developmental window. So that's kind of a wild card because it's not just a metabolite, but it's actually ensuring you have possibly a human-derived probiotic versus something from another source if it's going to be delivering a maturational signal.
Thank you.
Or just a quick, this is a methodologic question, so it's a simple one. Has anyone looking at the role of gastric transit time in this? Are there some bugs that swim upstream faster than others? And if you have a short transit time, how does that affect the overall populations? I'm always curious about that because obviously if you have diarrhea, you're going to be doing something. If you have constipation, you have a different role. And is anybody looking at that?
So the answer is yes. The microbiome that mostly we've been talking about is in the gut is the large intestine where the bacteria, they're there in huge numbers, but they're actually not stuck to the epithelial surface or the inner mucus layer. They're in the outer mucus layer and in the lumen. And issues of transit will impact on bugs that are present in the lumen and not so much those present in the outer mucus layer. That's in the colon. In the large intestine, in the small intestine, there's even more dramatic effects. But the only studies that I'm aware of they're very transient. So you see changes in bacterial compositions, but as soon as these are using drugs as prokinetics, it comes right back. So if you look at diabetic gastroparesis, where it's a more chronic condition, you see overgrowth of bacteria, but it's bacteria in the wrong place. They're sitting in the stomach rather than small bowel and colon.
Actually, this is an area that I don't think ever got explored well enough in the fiber period either, because when you read everything that fiber does, one of the things it can be doing is carrying certain types of plant compounds into the lower part of the small intestine and into the large intestine. So now you have a whole host of compounds that could affect the metabolism of that microbiome. And it's, in the early days of fiber, there were people trying to look at it, but the tools just weren't there. And it probably is an area that needs to be re-examined.
Thank you. Please.
If we could just get back to the topic of environmental exposure again. So I think it's been brought up that a lot of different things can impact the microbiome. And, it's certainly some of these things in the environment can impact it, but I'm wondering if it's too early to start looking at the impact of those things, because as the panel's already brought up, they're not validated biomarkers now to say, okay, is a change actually adverse or is it beneficial? So if you're going to start talking about, hey, there's an environmental impact on the biome, there's really at this point no way to interpret those changes as either being positive, negative, or non-impactful. So is the next step really defining what healthy microbiome means before we start looking at the impact of environmental exposure?
Well, I think when you're doing a lot of these large cohorts, and especially when you're making it a community resource, gathering that information and having it to be able to subsequently look at it, that if you're doing it, you want to get as much as you can and make it as much of a resource for the future as you can. In terms of defining a healthy microbiome, that's exactly what the American Gut Project is hoping to do. And it is also at the same time collecting all the environmental exposures to try and say then what influences the development of it. So I think that the two kind of have to go hand in hand and each provide parts of the puzzle.
So then is there a next step after that? So there's this exposure, you have an impact on the microbiome, but then isn't there still that next step of, are they biomarkers of actually impacting health or not? So a change in the microbiome is not necessarily indicative of positive change or a negative change.
Well, then I think the next step is then going back and really say what's different between health and disease, and can you modulate it to move on that spectrum, especially before you have avert disease, to move more towards health. So it's just providing the information to start knowing where to go in the future.
If I can add on that, I was on a program committee that was dealing with metals, particularly heavy metals and risk of infectious disease recently. And the entire program was diverted over to the microbiome because it turns out that availability of zinc, iron, arsenic, lead, cadmium is really so dependent upon the microbiome that you have to know what's going on there first in terms of what is sequestered, translocated, or made available from mammalian cell exposure or use, be it benefit or risk. And for that reason, we do need to understand how the microbiome is interacting with environmental factors prior to what it may do in terms of alteration of tissues or organs.
Thank you, Randy. Yes.
Thanks for a great afternoon. I'm thinking back to people talking about Paleolithic diets and diets from years and years ago. How has the microbiome changed? It seems to me, just thinking about those kids in different developing countries with big bellies filled with all sorts of parasites and worms, what's the microbiome like there? Because that's more, I guess it would be more typical of what humans have lived with for millennia than perhaps the microbiome today.
You want to start, or I can?
Start. So people have looked at the microbiota composition, not so much the function, of children around the world, and there's big differences, whether you're an urban dweller or a rural dweller. That's one of the studies that's been done in China. There's been a study of children in Burkina Faso who were undernourished, marasmus, not kwashiorkor, versus children living in Italy. Totally different diets, that's a confounding variable, but their microbiomes are totally different as well. There is the other thing that's a confounder is how much vegetarian, like if you're eating a plant-based diet versus an animal-based diet, because there's huge differences in composition in humans, whether they're primarily complete vegetarians, mixture, or heavy, I call it a Western diet with lots of beef in it. So all of those things affect microbiota composition. The part about the function, I'm not that clear on it. Maybe I'm not sure it's been done actually so far.
Yeah, the function really hasn't been done. Kind of one of the big buzzwords a couple of years ago in the microbiota literature was this whole idea of enterotypes, and that there were three main enterotypes, and this was mainly work by Gary Wu. And kind of depending whether you were eating a predominantly meat-based diet or a predominantly plant-based diet, it influenced the type of microbiota that you had. And subsequent work says that there is really a spectrum, but I think the very recent studies from Turnbau's lab definitely shows that there is an investment in the microbiota composition, and it's more to be able to utilize the food that's getting there for them to have the what they need. Different bacteria are there because if they can have what they need as nutrient sources. So that's part of why the shift is. The question is, what we don't know is just because you're having a shift or different bacteria, are they doing different metabolic functions, and is it having biological effects?
Rodney, any comments on that?
No, nothing to add. Thank you.
Thank you. Deanne?
Yeah, I just wanted to ask too, I know we're talking environmental and the polyphenolics, there's a lot of tannins and such that get to the lower GI tract and have been looked at a little bit in this area, but also Maillard reaction products, which gets you to cooking and processing and these sorts of things as well. Will that be part of the data collection as you see going forward and looking at different diets, not just vegetarian versus meat, but you know, effects of cooking sources and things? Because there's emerging data that anything, of course, any of these components that can get to the lower GI tract can either be fermented or have some effect on.
I know they're collecting information on complete dietary intake and stuff, whether it's getting to the level that they can measure the Maillard products and stuff like that, or if other people can estimate it from the food frequency questionnaires and the diet information they're getting. I can't really answer.
In Canada, I know you can do like genome-wide association scans. You can now do exposome-wide association scans. But how many things are actually included in that list? So that's the part I don't know either. Like, what level of granularity do you add to the list? Because if you ask people who are part of a cohort study to fill out these forms, and they take hours and hours, the forms don't get filled out. So it's always the balance between asking the critical question and not missing something. So as far as I know, the questions in the Canadian cohorts, which are children and the longitudinal study for aging, don't go to that level of granularity. But maybe they should.
Okay, folks, please join me in thanking our presenters today. This is